Hey everyone, so this video is about apoptosis. This is a continuation after we've just completed signal transduction. So our intention for this video is to know the purpose of apoptosis and to understand the two ways in which apoptosis is initiated. So apoptosis is programmed cell death. It's our way that our cells will actually maintain the cell numbers to make sure we're not getting too many cells. Or if cells are damaged or dangerous, apoptosis will actually cause them to die as well. So in apoptosis, it's using a series of different enzymes which destroy the protein components of a cell and start to break it down into smaller components. You can see here, this is a cell before apoptosis, and by the end of apoptosis, it started to break down into smaller fragments. So why do we need apoptosis? If we use apoptosis for a variety of different reasons. So for example, if a cell is infected, we would use apoptosis to make sure that that cell was going to die and not pass on the infection. If we have proteins that are not folding correctly, if there's a mutation in that cell, we'd use apoptosis to destroy that cell. Or if there was some other damage to the DNA, for example, through radiation, we use apoptosis to control those cell numbers in a programmed way. So there are a couple of key terms and components you need to know about for apoptosis. The first are these enzymes called caspases, and these are protease enzymes. What that means is they break down proteins, and they're really important in apoptosis. And what they do is they actually go and they attack specific proteins and break them down. And the term we're going to use is cleave. So in this video, we use this star symbol as a symbol of our caspases. And you can see they're going to move and cleave specific proteins down. The second term is you're going to hear this term called the caspase cascade. This is where one caspase will actually activate other caspases, and like a chain reaction, we can have one caspase activates multiple others. And this just means that one signal can actually amplify the apoptosis process. It makes it into a really big reaction, just starting from one caspase being activated. So that's called the caspase cascade. So in apoptosis, there are two ways in which apoptosis occurs. One is called the extrinsic pathway, and if we know this X means it's come from outside the cell, so that's where something has told the cell to commit apoptosis. And then we've got the intrinsic pathway, which is where the signal is coming from within the cell. So extrinsic means the signal is being sent from somewhere else, and intrinsic, it refers to the signal coming from the inside. So the extrinsic pathway works with a ligand, and we know that ligands are the signaling molecules. The specific ligand we're talking about is called the Fas ligand, and the Fas ligand binds to the receptor. In this case, it's called a Fas receptor. That activates a series of secondary messengers. The specific one is called the Fad protein, which then activates a caspase, and we know that they're the really important enzymes that start breaking things down, and those caspases then start activating each other using the caspase cascade. So just in summary, we have a ligand, a signaling molecule, binds to an extracellular receptor, which causes a secondary message, which then causes the caspases to be activated, and therefore the caspase cascade. It's an example of signal transduction. We had reception, transduction, and response. This is used a lot by the immune system. You can see in this GIF, here's an immune system cell. It's got the Fas ligand, which binds to these Fas receptors. And that immune system cell is saying to this cell, hey, you need to commit apoptosis and destroy yourself. So again, there's our binding of the Fas ligand. That causes a secondary messenger or a chain reaction. So here we've got our Fad protein. And that Fad protein is then going to activate a caspase, which we then know goes and activates other caspases in the caspase cascade. And they're just going to go and break down the proteins in a cell. So an example of the extrinsic pathway would be for the immune system. These are cells trying to destroy infected cells. The second pathway is the intrinsic pathway. And this one's a little bit more tricky. It relies on having two proteins in balance. We need to have the same amount of protein 1 and protein 2. Well, normally there's a balance, but if there is damage or some problem with the DNA, that balance is lost. And I'll explain it with this diagram. So these are our two proteins, and as you can see, normally they're in balance. They cancel each other out. This anti-apoptotic protein is trying to stop apoptosis, and this pro-apoptotic protein is trying to undergo apoptosis or start apoptosis. So normally they're balanced, which means there's no apoptosis occurring. But if there is some sort of damage, that balance is lost, and this pro-apoptotic one actually goes and you can think of it like drills a hole inside the mitochondria. And what that does is the mitochondria then release this protein called cytochrome C. And cytochrome C goes to another reaction with another protein. And eventually it activates a caspase, which then goes and activates the caspase cascade. So in the intrinsic pathway, we have two proteins balanced. When it's lost, one of them cuts a hole, the pro-apoptotic drills a hole in the mitochondria or damages the membrane of the mitochondria, which releases cytochrome C, which eventually activates a caspase and the cascade from there. So what that causes is the caspases then start cleaving or cutting specific proteins, and the cell begins to shrink. And we also have this term called blebbing. So here you can see this cell is just going about its normal business. Then it's either received an extrinsic signal or an intrinsic signal, and it's starting to now activate the caspase cascade. You can see it's starting to shrink, and then we have these things called blebs or fragments starting to break apart, and then these get engulfed by another cell when they're done. So eventually we'd have small little fragments that get engulfed by other cells known as phagocytes. So that's the general basis. Now, sometimes apoptosis can go wrong. So if we don't have the exact right amount of apoptosis, if we have too much or too little, it can actually really start to damage the cells in the body. So if we have too much apoptosis, the cells could actually start committing apoptosis in cells that are not damaged, that are needed. And so something like Alzheimer's, where there's breakdown of brain cells, that would be caused by an overactivation of apoptosis. The exact opposite is if we didn't have enough apoptosis, we could have cells starting to divide uncontrollably. So cancer is a really good example. Normally, we would cause apoptosis, you wouldn't have the cancer occurring. So here's a cell with some damaged DNA. When we conduct apoptosis, there's no risk. However, if apoptosis didn't occur, we can see that cell will continue dividing, and we then have a much greater chance of developing cancer. So in review, we have two different pathways, the extrinsic and the intrinsic. The extrinsic relies on the Fas ligand, whereas the intrinsic is about those two proteins that are balanced. They involve caspases. Both of them result in the activation of caspases, and they are enzymes which cleave proteins. And we know that caspases can cause other caspases to be activated, and that's called the caspase cascade. And lastly, those caspases cause the shrinking and the bleeding of a cell. So for this video, you need to get your head around the two different pathways, the extrinsic and intrinsic pathway. Hope this video helped. Thanks.